Dear students, now we are going to see last part from our chapter meiosis. Last lecture I have taught you mitosis. There are four phases: prophase, metaphase, anaphase and telophase, and how the cell divides. Now here in meiosis you can see only in somatic, mitosis you can see in somatic cells, meiosis we can see in germ cells. So it is for sexual reproduction, germ cells, or you can say in sex cells or in your gametes. Let us read the point from your textbook. Meiosis is completed through two stages. There are two stages for meiosis, and those two stages are meiosis 1 and meiosis 2. So two stages of meiosis are meiosis 1 and meiosis 2. Two phases are there. In meiosis 1, what happens in meiosis 1? There is recombination or crossing over between homologous chromosomes. You know, homologous chromosomes means same chromosomes. And then the homologous chromosomes are divided into two groups. They are divided into two groups, and two haploid cells are formed. Two haploid cells are formed. Now it is not too much in detail for you. So see here, meiosis 1. In prophase there are 1 to 5 stages, that is liptotin, zygotin, pachytin, diplotin and digynesis. Then second phase is metaphase, anaphase and telophase. In meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2. So here you can differentiate it. In meiosis 1, prophase 1, then metaphase 1, anaphase 1, first phase it is, and telophase first. In meiosis prophase 2, it is second stage, so it is named as prophase 2. Then metaphase 2, anaphase 2 and telophase 2. So thus these are these stages in which the meiosis takes place. Then here meiosis 2 is just like mitosis. In this, the two haploid daughter cells formed in meiosis 1. Now here in meiosis 2, what happens? In meiosis 2, it is like mitosis. You have seen in mitosis 4 phases. In the same way here also 4 phases are there. Two haploid daughter cells. Haploid means n number of chromosomes are there. Two haploid daughter cells formed in meiosis 1 undergo division. They divide, and what happens here? By separation of recombined sister chromatids. The sister chromatids which are formed in meiosis 1, they undergo division, and 4 haploid daughter cells are formed. 4 haploid daughter cells are formed. Then process of gamete production and spore formation occurs by meiosis. So gametes are formed by meiosis, spores are formed. In this type of cell division, finally you get 4 haploid cells from one diploid cell. Means from 2n we get 4 cells, 4 haploid, where haploid number of chromosomes are there. Due to this, all 4 daughter cells are genetically different from parent cells. मग ते ह्यांचं जे जेनेटिक्स असतं, जेनेटिक मटेरियल, त्याच्यापासून हे चारही सगळे कसे असतात, वेगळे असतात. जेनेटिक मटेरियल इज डिफरंट. नाउ सी देर इज नो डायग्राम फॉर यू इन दिस meiosis, but then also one figure is given in 11th standard book that I will explain you how this meiosis takes place. Means you will understand. Suppose this is a diploid cell 2n. Then here one is maternal chromosome, one chromosome is from female, that is maternal chromosome from mother, and one is paternal chromosomes. Now they go duplication of chromosomes. These figures are not included in your textbook. I am just explaining you it, referring the textbook of 11th standard biology book. So here they duplicate. Means this single, it becomes double. Here also same thing. So here you can write each chromosome. See here, each chromosome has 2 chromatids. Each chromosome has 2 chromatids. Now they again divide, and this is meiosis 1. What happens in meiosis 1? So here you get combined cells now. And here the second stage is meiosis 2. Again these two cells divide, this cell divides, and here you get 4 haploid. Meiosis 2. Once again I am telling you, this figure is not there for your studies, but only just to understand I am explaining you how the 4 haploid cells are formed by 2 stages of meiosis. Mitosis only one stage is there. Mitosis मध्ये फक्त 4 फेज असतात, मेटाफेज, एनाफेज, टेलोफेज, प्रोफेज, मेटाफेज, like that. Here meiosis 1 4 phases are there, meiosis 2 4 phases are there, and from one diploid chromosome, from one diploid cell, we get 4 haploid cells. So let us see. These are 4 haploid cells. So n number of chromosomes. This is meiosis. What happens in meiosis? Meiosis first and meiosis second, that is given in this figure. This is not for your, in your syllabus it is not included, but to understand this meiosis I have explained you how meiosis 1 and meiosis 2 occurs, and the diploid cell gives us 4 haploid gametes. Now there is one question in your textbook. That question is, with the help of suitable diagram, give 5 stages of prophase 1. We studied in meiosis 1. Meiosis 1, first stage of meiosis, there are 4 phases. First, prophase. Second is prophase 1, then metaphase 1, anaphase 1, and telophase 1. Prophase 1, there are 5 stages in this, and which are they? They are liptotin, then zygotin, pachitin, and fifth is dikinases. Now your question is, with the help of suitable diagram, explain the 5 stages. Explain the 5 stages of prophase 1 of meiosis. What we have to explain? 5 phases, 5 stages of prophase 1 of meiosis. So how will you start your answer? Prophase 1 of meiosis is much longer phase of the meiosis. It is subdivided in 5 substages. First, liptotin. Second, zygotin. Third, pachitin. Fourth, diplotin. And fifth, dikinases. Now we will study each in detail. So first let us see liptotin. Now what is the meaning of liptotin? Liptos means thin and nema means, or you can say here liptonema. Liptotin or liptonema, and nema means thread. So meaning of liptotin is thin threads. Now what are these thin threads? So initially the chromosomes start condensation. You know that chromosomes are thread like structures which we can't see clearly, but in this stage initially the chromosomes start condensation and they become compact during liptotin. Compact means here in figure you can show. Now for this each stage there is a figure. So here centroid is there. This is central. Then chromosomes are shown. You can see the chromosomes in this stage. Then here it is nucleus. So let us label. These are chromosomes. Then this is centromere. Then nucleus membrane. Nuclear membrane, and this is nucleolus. Nucleolus. Then second phase is zygote. Zygote or zygonema. Now what is the meaning of zygo? Now you have zygo means it is pair, and nema means thread. So threads are getting paired in this case, and the homologous pair of chromosomes start combining here. So what will you write here? In zygote, homologous chromosomes start pairing. This pairing is called synapsis. It may come for MCQ. The pairing of the homologous chromosomes is called synapsis. The structure called synaptonema. Synaptonemal complex develop twofold chromosomes in place during pairing. Each chromosome's chromatid arm divides and forms structure called bivalent or tetrad. So see how is zygote in. Let us see what is tetrad or bivalent. These are centrioles. So this is a bivalent or it is also called as synaptonemal complex. Then this is centromere, and these are homologous chromosomes. The homologous pair of chromosomes. This is nucleolus. So this is zygote where a bivalent is formed. Figure B, zygote. Third stage is patching. Now what is the meaning of patching that we will see? Patches means thick and nema means thread. So here this thread start become thick. You can say patchitin or patchitinema. And what happens, let us see here. I will draw first figure and then I will explain you. Central starts moving. Central is moving and this it moves to ends, and here the threads become thick and also they start crossing over. So this is crossing over in homologous chromosomes. In homologous chromosomes. And here nucleolus starts disappearing. Disappearing nucleolus. So this is pagetin. Now in this description what you are going to write? Crossing of sister chromatids of homologous chromosome then genetic recombination. Now see here, as there is crossing, the genetic recombination is produced here. Genetic recombination is produced due to such exchange. The homologous chromosomes remain